All right, so I'm going to talk about one concrete project. As Kevin already alluded to, it's sort of going into the direction of people using data about themselves rather than looking at other people's data and other people's project. It's sort of making a project out of yourself. And I'm going to, I don't really know which screen to look at here, but the project is at this interface of three things. Taste and smell is sort of the topic. We're going to use personal genomics as our research tool, and I'm also going to talk about informational self-determination. That's the My Data project that Kevin alluded to. And we are just getting started with this project, so I'm not going to tell you any results that we have, but here's sort of the outline of the project. We want to recruit 200 to 500 citizen scientists from the Zurich area. Most of them will probably be scientists because we will try to recruit people at the university, but they may be from other topics, not necessarily bioscientists. So they're sort of scientists and citizen scientists at the same time. We will get ourselves a genotype. We'll talk a little bit about that. So we'll have our genomes analyzed, and then we'll use that information to find the genetic drivers of our inter-individual variation in smell and in taste perception. And this is clearly in Switzerland certainly a new type of project because the use of personal genome data is something that's quite heavily regulated in Switzerland. There's several laws that are governing this, and we don't fit in anywhere at this point. But I think fundamentally we all should have the right to use our own genomes to do with them whatever we please to do. And so we will have to sort of sort out some of the surrounding issues here, and that will be a good project to do this. And the goal here is that we all together learn about the ethics law, philosophy behind personal genome data. So having people from lots of different backgrounds in this project will be very interesting. And it will also be sort of a beta test for this personal health data cooperative platform that we founded. So here is sort of like we can do a citizen science project here right now. Most of you probably know this. When you eat asparagus, afterwards your urine smells. But, so who doesn't smell this? Usually it should be, okay, there's 1, 2, 3, okay, so usually it's about 7%, 7%, so 1 in 15 cannot smell this. And they can smell everything else just fine. But not this one. And so the reason is there's this compound, this asparagusic acid, which breaks down into these compounds and they smell, and these 7% of the population just cannot smell this. And so this is sort of like a famous example, but there are a lot of these things actually. There's violet, there's little flowers. It's sort of like hand soap. A lot of cheap hand soap has this smell, beta-iodine in it. And look at this distribution here. So there are people, this is 6 orders of magnitude. Difference in concentration, that's the detection threshold where different people can pick this up, okay? And so this is sort of like, if you would have that with vision, you could see, you know, I don't know, read a newspaper on Mars or something. The difference is very dramatic. And there are a whole series of smell compounds, grassy smell, cheese smell. So these are sort of the individual compounds in here that have been tested that give you this smell. And we have this large distribution in the sensitivities, and you see very often you have these bimodal distributions, okay? And these all happen to be genetically driven, that's already known. So how does this work? How does our nose know? So we sniff air in, and then it gets sort of to this olfactory epithelium, and in this cells you have these olfactory receptors that sit on the cell surface, and the smell compound binds to it. So this is a picture of one of these receptors. I don't know if this was done with folders that people did, no GPCRs. Okay. So what basically happens is a little odorant molecule comes in. It's sort of like, if it fits perfectly in this binding pocket, then it triggers a response in this nerve cell, which then goes on to the brain. And there are about 800 olfactory receptor genes in the human genome, but only 300 of them are active. But different ones are active in different people. Okay? And you have a very large number of genetic variation in these receptors on top of it. So you have this enormous genetic diversity, which corresponds to this diversity that we have in our perception. And so, general sort of primer biology 101 here, genetic diversity in humans. We have 3 billion base pairs in the human genome. We have 2 copies of the genome, one from the father, one from the mother, and greater than 99.9% of the sequence between 2 individuals is identical, but that leaves more than 40 million variations that have already been cataloged. And these variations are dominated by what's called a single nucleotide polymorphism. And so this is what this is here. If you have the sequence of your genome of one person, of another person, you will find that there are certain places where one of the letters is swapped. And so what happens is that this genetic information is being transcribed. You may make a different receptor here, and then as a result, that receptor may work differently in one person than the other, and that may make you tall or short or fat or skinny. So that's basically how this the process works, how genetic variation influences our phenotype, how we are. So the quite amazing thing that has developed in the last few years, we saw sort of like the speed of Moore's law, how things are developing very quickly. So the speed with which we can analyze genetic information is moving or is increasing much faster than the speed of computing. So we also have this sort of problem that the computers can't keep up with the genetic information that's being accumulated. And so we can now very cheaply in the lab analyze the human genome for about 200 francs. If you want to do the whole sequence, you're at 1,000 Swiss francs or $1,000. This technology is really quite amazing. I don't want to go into the details, but it's really a solved problem. The issue is not that it's still technically challenging. Everybody can have the genome analyzed. It's sort of a question of who gets access to this information. I'll talk to you about this in a second. So when we have now our genetic information, we can sort of like go the other way around and try and find what genetic variation is causing this phenotypic variation. And there's this technology called the genome-wide association study, sort of a statistical method where you can sort of look at variation in the phenotypes. Is somebody tall? Is somebody short? Can you smell something or can you not smell it? And from that, you can go back and you can find the genetic variation that's causing this. That's basically what we're trying to do in the Citizen Science Project, is to try and find the genetic variations that are causing this difference in the perception, right? Why can I smell something and somebody else cannot smell it in places? Okay? And so basically all you need to do here is you have the individuals and you need to phenotype them and you need to genotype them and you put them together, the two pieces of information, you kind of turn the crank, and then what it will spit out is what's called a Manhattan plot, because it sort of looks like a big city with skyscrapers. And the peaks here are places where there's a strong statistical association between the genetic variation that you carry and the fact that you have a certain phenotype. So for those who are not so mathematically inclined, here's sort of an example. If you have people here who have a green head and people who have a red head, and you want to find out what is driving this genetically, what you would do is you look at their genome and you look for places where the fact that they have a green head sort of associates very strongly with a particular genetic variation. And because the phenotype is not driving the genetic variation, you kind of know the direction of causality here. And so you can say, okay, that place in the genome has something to do with this particular phenotype. And you can do this for anything. That's the beauty of this project in a way. Once you have the genotype, you can get together with a bunch of other people and say like, hey, I wonder why people have done this for like earwax, the type of earwax they have, because there are two different types. What is driving this? You can find out. People have found out. And so this is the cool thing. Once we have this genetic information, we can do this kind of analysis, and people can decide what interests them, what they would like to know about themselves, and they can do it themselves. This is one project I'm going to maybe try to get people involved in tomorrow in this hackathon to build something, a platform that people can bring their own genotype and then ask questions and do a GWAS study. So here are sort of some results of this kind of work from a previous study that I did in my previous job from 600 people. And actually the first author is somewhere here, Mirko, where is he? Sitting in the back. There are, this was on taste, not on smell, so we found a genetic location which actually was known previously already. It's a classic one. It's more of a validation thing. This compound is related to the compounds that make Brussels sprouts and broccoli taste bitter to some people, and you can sort of nail down this location on chromosome 7, which is actually a taste receptor. And there's a polymorphism in this taste receptor. We also found for the first time quinine, which is one of the bitter compounds in tonic water. It's a different bitter taste receptor on chromosome 12, and also for caffeine in coffee. Even though the bitterness of coffee does not primarily come from caffeine, but I thought I'd show a nice picture of a cup of coffee. And there again is a location actually quite close to this one that's causing this variation. What you see is that sort of this one is getting kind of noisy here, right? So you have this very large signal here and smaller signal, and that is because the genetic variation does not have such a strong influence in the case of caffeine on the perception of bitterness as in the case of protein. So in some cases, if you have a very large genetic effect, you can find these associations or pinpoint the underlying genetic variation very quickly or very easily. Now, the one case here, the asparagus, people have done this also, and this was a study done by 23andMe, and they found the genetic location there, and this sort of brings me to 23andMe, which is sort of like about digital self-determination and sort of who owns my genome. So this is something from the privacy statement of 23andMe and the informed consent statement. And basically, if you go get yourself analyzed at a company like 23andMe, they have the similar conditions as you would have user agreement signing Facebook or with Google, which basically is that they own everything that's in your genome. They can make commercial uses of it, they can use it for any kind of study, they can sell the information, and so on. And this is sort of something that is quite general problem that we as individuals, we generate a lot of data, and this data goes into all sorts of places. And after that, we're sort of a bystander, right? Our data goes here, and the places where then the exchange of the information takes place is very often the places where we don't want this to happen, okay? And the places where we want it, between, you know, the hospital and our doctor, there it doesn't work, okay? So it's sort of like, I don't know if there's like a Murphy's Law equivalent. Typically, when you want it to work, it doesn't work, and when you really don't want it to work, it's probably taking place. And I can sort of skip over this. So, one of the things that we're trying to do in an ETH-sponsored project is to actually change the situation and actually put the individual in control of their data. And the mechanism we think will be the best for doing this is to actually have a cooperative that's owned by individuals, and you can put your data, medical data is what we're going to start with, you can put your data into this cooperative. The data is encrypted. You only have the key, but you can make this data accessible to people who want to use it for projects. You can share it with your doctor, but you can also, anytime you want, you can retract that right for somebody else to look at your data. Okay? So, in the to kind of try to stay on time, here is sort of a more detailed view of how this project will work. There's sort of a central database with where the data is kept under lock and key, and you can have access to it. You can import your data, and the data can be analyzed for research projects. And the idea is that the analysis comes to the data and not vice versa. Okay. Here are some of the other people. I heard there were people from the Fachhochschulen here, so we actually have Serge Vignon from the Berner Fachhochschule, who is in this project. Ernst Hafen, the former president of the ETH, and several other ETH professors are involved. So we have an illustrious crew of people who are driving this project. Okay, that was it, and I think questions will come afterwards.